The nucleus is an example of an organelle. It is specialized to contain the genetic information the cell needs to do all the other things it's going to do. I don't want to dwell a lot on this, but let's just kind of take a look at some of these others that are shown here in the picture. I'm sure you've noticed this great big thing that looks like a garden maze or a corn maze that's been painted orange and blue. Probably a Gators fan, right? So he's like, I don't believe she said that. Yeah, I did. All right. This big structure here is something called the endoplasmic reticulum. You know, I haven't done this for a while, but a few years back, some of the other faculty and I sat down, and we figured out that between your tuition, your fees, your lab fees, textbook costs, and all of that, how much money it cost you for each time you came to class. So we broke down the whole total across 15 weeks and 2 class sessions and a lab each week. And I don't remember what it was at the time, but I think we figured out that it cost you about $40 for every class, right? With adding in the book, which is ridiculous, right? So every time you come to class, you should be getting $40 worth of education, right? All right. This is at least a $10 word. Right? Endo means inside. Plasmic is referring to the cytoplasm, so it's inside the cytoplasm of the cell. So we refer to something as reticulated when it is linked together or chained together or forms an elaborate woven network. And so the endoplasmic reticulum is in fact a fairly elaborate linked together network of cell membrane. Here's a close-up picture of this. Now, we really didn't get much of an idea about what was going on here until we developed the electron microscope. This is virtually impossible to see with a normal light microscope. It's very small and it's very thin. But with the higher magnification you get with electron microscopes, not only could we see this network of membrane, this thing that looks like a garden maze, but we also saw that it had 2 different sections that looked visibly different in the pictures. There's a section of the endoplasmic reticulum.
Sorry, I wasn't trying to shake you.
No, I was just wondering what you were doing.
Oh, because my ink is like drying up.
Oh, I'm so sorry. And that's your only pen.
Except a red pen, but no, you're okay. Just continue.
I don't know. I'm a fixer.
Now you can make me feel bad.
That was a freebie giveaway. Let's make sure it works. There we go. All right. So you don't have to give it back. You can have it. It was a throwaway. All right. So we have this one area that looks rough, gritty, like a pebbled, sandpapery kind of surface. And not surprisingly, this is referred to as the rough endoplasmic reticulum. But other sections of the endoplasmic reticulum don't have that same gritty, sandpapery look. The membrane here is smooth and featureless. So that is referred to as the smooth endoplasmic reticulum. Now, both of these act for the cell as a kind of a work surface. When you guys have got a big project, something that's going to take a lot of steps and a lot of effort, probably the first thing you do is you clear off your the kitchen counter, or you clean off the workbench in the garage so that you can set everything out where you're going to need it and can get to it easily. So Your kids come home and they say, we need 12 dozen chocolate chip cookies for the bake sale tomorrow, Mom. We forgot to tell you last week. Okay. So you move everything else to the side. You get out the flour and the sugar and the eggs and the butter and the chocolate chips and all the vanilla, and you put it all out there where you can find it. It's a work surface. Well, that's what the endoplasmic reticulum is for the cell. It's a work surface. It's a place where it can do complicated things and keep everything where it needs to be so it can get to it quickly and easily. The rough endoplasmic reticulum is a place where the cell makes proteins. In particular, it is making protein molecules that it is going to secrete out of the cell. Substances that it is going to push out into its extracellular environment. These might be things like hormones, growth hormone, insulin, prolactin. It might be neurotransmitters, chemicals that allow cells to communicate with neurons to communicate or muscle cells to communicate. But this is a specialized area where it's going to make these kinds of proteins. And that's the reason why it looks so gritty. Because covering the surface of the rough ER are thousands and thousands of little structures that are called ribosomes. Now, we're not going to go into the whole protein synthesis thing. We're not going to be talking about transcription and translation. Yeah. Some of you are groaning. You remember that from bio, all right? But ribosomes are the actual structure that make proteins. And they're all over the cell. Your cells have hundreds of thousands of these guys. Some of them are attached to the rough endoplasmic reticulum, which was what makes it look rough. Those are the ribosomes that are making proteins for secretion. Ribosomes in other places are making proteins for other purposes. You're looking confused. You're not confused? All right, good for you. All right. Now the smooth endoplasmic reticulum is smooth because it doesn't have any ribosomes. So all we're seeing is the membrane. This is also a work surface. Just wondering what you're up to.
I'm trying to get the shoes on.
All right. We have water and paper towels right there. Oh, how about that? All right, you don't get to escape that easily. Yeah, I'm just going to go down to the ladies' room and wash my hands. No, you're going to wash your hands right here. Okay. There's no escape. When I get to talking, everybody has to listen. Now, the smooth ER doesn't have any ribosomes, so that means this is not a place where the cell is making proteins. The smooth ER is the place where the cell is making lipids. In particular, this is a place where it is synthesizing steroids. Now, if you're not too sure what the difference is between proteins and lipids, you can go back into the book and look at chapter 2. Which is about biochemistry, but once I get sucked into that, I'll never get us back out. So I'm not going to try to give you guys a lesson in biochemistry. There's also the first lab in the lab manual. It has a shorter version of the difference between proteins, lipids, carbohydrates, and nucleic acids if you want to read through that. But lipids would be the area of the fats and the oils. It also includes waxes, cholesterol, and the steroids. And steroids are used in a lot of places in our body as communication chemicals. So some of our hormones are steroids, things like cortisol and aldosterone, progesterone, testosterone, estrogen, all of those are steroids. All of those coordinate activity between different parts of the body. Steroids are also used in chemical signaling for things like body temperature regulation, Some of them are used to stimulate smooth muscle contraction. So steroids are found in performing a lot of different functions in the body. They get made at the smooth endoplasmic reticulum. Now, over here, this green thing, and remember it's not really green, right? This green thing here is an organelle called the Golgi Body. Some textbooks call it the Golgi Apparatus. And some books simply call it the Golgi. Now, it's capitalized because this was a guy's name. There was a hair professor, Dr. Mr. Golgi. So it does have to be capitalized because it's referring to somebody's name. I don't really have a great close-up picture of this. I think this is the best one I've got. All right. The Golgi apparatus, the Golgi body, is usually found fairly close to the rough endoplasmic reticulum. Proteins that are made at the rough endoplasmic reticulum are then transported to the Golgi. The Golgi body, the Golgi apparatus, is a place where these proteins are then modified prior to secretion. Anybody here ever build models, model airplanes, model ships, model cars, right? Model trains, nothing? I got one nod, all right? I will talk to you then, all right? You know that when you first get the pieces out of the box that they've got little flanges of plastic on them and you got to kind of trim that off and file them down and you know, so it's not going to be a perfect fit until you get it reshaped. You have to do a little modification to the pieces that came from the factory. It might make a little more sense to those of you who don't do models to think about when you buy new clothes, you got to trim off the tags before you wear it, otherwise people laugh at you, right? Walking around with a big price tag hanging down off your sleeve, you got to take that off. And the security thingy and the, you know, you got to take all that off. Well, when proteins are first made, they've got little extra bits and pieces like that that work to tell the cell this is a secreted protein, move it to the Golgi body, but then you got to take some of that stuff off because you don't need it anymore. So one form of modification is called proteolytic cleavage. Proteo, of course, is referring to proteins. Cleavage means to cut something off. We're not talking about boob cleavage, all right? Cleavage typically means to separate, to cut through. So we're going to cut off little pieces of the protein that we don't need. We're going to trim it down so it'll be just right. Lytic also means to break or to cut. So it's a little bit redundant. Proteolytic cleavage, we're cutting off little pieces of the protein. Now, other proteins may not need that kind of trimming, but they may need other types of modification. One other form of modification we see with some proteins is a process called glycosylation. Glyco means glucose, which is a form of sugar. So glycosylation is putting little pieces of sugars on the protein. It's kind of like bedazzling your clothes. You're going to make them all kind of sparkly. And again, the shape of the molecule will determine the function of the molecule. Adding the sugars adjusts the shape just a little bit so the protein will have just the right shape. It also can act as a kind of a this end up signal. But putting the sugars on one side tells the cell which way the protein is supposed to face. And third, there is phosphorylation. Now, what I've drawn for you here is a quick and dirty version of what a phosphate group is. The P stands for the element phosphorus, the O's are oxygen, and the H's are hydrogen. This whole little unit is called a phosphate. They're very important in biology. They play a lot of different roles in physiology. One of the things that they do is they act as an on-off switch for protein activity. Some of our proteins do things. They don't just lie there forming structures. They're enzymes, they're catalysts, they build things up, they break things down. They're active molecules. If you put a phosphate or phosphates on a protein, that's like flicking off the on switch. And now the protein will be active. Take off the phosphate and it's the off switch. Without the phosphates, the protein loses activity and doesn't do all the things we want it to do. So for proteins that are going to be leaving the cell, we want them to work when they get there. Right? Remember Christmas? And you had to buy batteries to go with the toys that you were giving your kids? Because Christmas morning, they get the toy and no batteries to make it work. They're going to be very, very unhappy. Right? So by adding on the phosphates before you send it out of the cell, you're making the protein functional, active, and ready to go. So some of our proteins that must be phosphorylated, all of these things occur in the Golgi. So depending on what kind of protein it is, what kind of molecule we're working with, it will either be cleaved, trimmed, glycosylated, or phosphorylated. And yes, you can add that up. So they can be cleaved and phosphorylated, they can be cleaved and glycosylated, they can be glycosylated and phosphorylated, or you can do all three. Depends on the molecule. After all that, the proteins are pretty much ready to leave the cell, and they will be exported to the outside. Now, these little orange guys, you see there's a lot of these in the cell. These are the organelles that are called mitochondria. Big picture of this one here. Mitochondria are also organelles. They are made of membrane. Most of our cells have somewhere between several hundred to several thousand of these organelles inside them. The mitochondria are the main location for an elaborate metabolic system called cellular respiration. Those of you who had biology either in college or in high school are now wincing at the memory of cellular respiration. This is going to come up later when we talk about muscles. So I can't avoid this altogether, but now is not the time or place to go into a detailed explanation of what this process is. Essentially, it is a way for the cell to capture energy and to store the energy that it captures into a particular type of molecule. Cellular respiration is going to take some sort of nutrient molecule, something that we would consider food, and break that molecule apart piece by piece, ripping apart molecular bonds and releasing the energy. And as it does that, it tries to grab that energy and transfer it into these molecules of adenosine triphosphate, or the abbreviation ATP. Now, ATP is an extremely useful molecule. These guys are essentially the batteries of the cell. Anywhere the cell needs energy to make something happen, it's going to use molecules of ATP. Just the same way, anytime we've got something we want to make happen, we put a battery into it. So your phones run on batteries, and your watches run on batteries, and your laptops run on batteries, and your car starts by using a battery, and your flashlights work with batteries, and the radios work with batteries, and the tape recorders work with batteries, and everything works with batteries. Anywhere you need to make something happen, anywhere you need to put energy into the system to make it happen, to make it go, you're using batteries. The cell has the same approach, but instead of Evereadys, Duracells, it's using ATPs. Anywhere it needs to put in some energy to make something happen, to move something, to break something down, to build something up, any place that's going to need energy is going to use molecules of ATP to supply that energy. To keep that energy flowing, you go to the store and buy more batteries, or you plug into the wall to recharge your batteries. The cell makes more ATP through this long process called cellular respiration. So typically it will start with something that we would consider a nutrient like glucose, a simple sugar, and it's going to break it down, 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 until all we're left with is carbon dioxide and water. What do we do with the carbon dioxide?
Exhale it.
We exhale it, we breathe it out, it's a waste product. What do we do with the water? You don't breathe it out necessarily, but yeah, you'll pee it out. You'll get rid of it. All right. So these waste products can be eliminated from the body very easily. But in the meantime, the energy that is being released is being used to make molecules of ATP. That is primarily occurring in the mitochondria. The more active the cell or the tissue is, the more ATP it needs, the more mitochondria it's going to have. Now we have structures that don't technically meet our definition of organelles. These cellular structures are not made of membrane, they're not enclosed in membrane. Doesn't mean that they're unimportant, it just means they don't qualify as organelles. Ribosomes would be one example. These structures are collections or aggregates of molecules of RNA and protein, but there's no cellular membrane covering them or enclosing them. They make protein molecules. Another group of structures that don't qualify as organelles are the structures that form the cytoskeleton. Said that like it was like, ooh, cytoskeleton. I kind of think it is woo. That's as good as that's going to get. All right. It's hard for you guys to see in the projection. When you look at the pictures in the book, it's going to be a little bit clearer. It just doesn't show super well with all this light. But if you look between the organelles in the cytoplasm, you see that it's not homogeneous goop, that there's actually smaller structures there that are organizing the cytoplasm. Those structures form a whole collection, a whole network called the cytoskeleton. It's composed of microtubules and protein filaments. Now, a big part of what the cytoskeleton does is simply help the cell maintain its shape. Just like your skeleton makes your body maintain its shape by giving it support, holding it up, the cytoskeleton microtubules and filaments also help the cell maintain its shape. They support the cell, they keep it from just collapsing and oozing away. But in addition to that, we've discovered that these microtubules and filaments can do some other interesting things. For example, parts of the cytoskeleton act as a kind of a guidance system for when the cell moves things from one part of the cell to another part of the cell. So if the cell is going to move proteins from the rough endoplasmic reticulum to the Golgi body, how does it know where to take them? How does it know how to get there? Why don't they just wander away? Well, it turns out that the cell uses these microtubules kind of like the rails on a railroad, and it will run these molecules along the tubules to get from point A to point B without getting lost or confused. So it's working as a guidance system, a railroad system for the internal movement of this and that. All other elements of the cytoskeleton allow the cell to alter its shape temporarily and in specific locations. I'm kind of jumping ahead here, but the perfect example of this would be when cells engulf bacteria, right? A lot of your white blood cells can do that. So, you guys know I can't draw. So this is your white blood cell. And there's a streptococcus bacteria that could give you strep throat and rheumatic fever and mitral valve prolapse and all kinds of horrible things that you don't want to have. So these white blood cells have the ability to kill the bacteria by pulling it inside. So the white blood cell will form this in pocket Using its cytoskeleton, it'll draw down a region of its surface, pulling down on the bacterial cell as well. And then these edges will come up and around until it has completely surrounded that bacterial cell and brought it inside. And now it can kill it using digestive enzymes and caustic, toxic chemicals. It can kill it and destroy it and make sure it never hurts you again. Yay. But in order to pull down this area, that's going to require activity by the cytoskeleton. And so these microtubules and filaments will actually work with each other to pull down that pocket to perform this kind of activity. It's changing the shape of the cell temporarily and in a specific location. We also have a couple of structures that are specialized for movement. Again, those of you who had some bio in either in college or back in high school, you guys, this is going to sound familiar. The two structures that we find in human cells are the cilia and the flagella. These are essentially extensions of the cell that through the microscope look like little hairs sticking out of the cell. Like that. Now, their internal structure and the way they work is very, very similar. Flagella, however, tend to be long. And cells that have flagella usually only have one, or in the wider biological world, maybe two or three. Right? Humans only make one type of flagellated cell. I just drew it. You recognize it? Sperm, yeah. That's the only type of cell we make that has flagella. Cilia, on the other hand, tend to be fairly short. And cells that have cilia tend to have a lot of them. They still look like little hairs. And in both cases, they work by bending or whipping. And I've got a picture of that. Now, this is the one from your textbook. And it's a very nice picture, but it's a little on the small side. So I have a bigger picture. Cilia, as it turned out, have a kind of a process for creating motion that's very similar to human beings doing the breaststroke when we swim, right? So the cilia sticks out straight, and then it sweeps down, moving the fluid, and then it curls back up to recover to the straight out position. So just like doing the breaststroke, the cilia move by being rigid and coming down and then curling back up into position. Now, in biology, you might have seen microorganisms that use cilia to move around their world. I think paramecium are really cool. Those are the ones that look like little flying bedroom slippers. Right? They're really kind of neat. We don't do that. Our cells don't do that. We don't have any cells in our body that move themselves with cilia. These guys are not for locomotion. What we get are the cells moving the fluid around them using cilia. The cells are going to stay put in the tissue, attached to other cells. But by moving the cilia, they can move the fluid around them. One really good example of this is seen in a system called mucociliary transport. There's another $10 word. Mucociliary transport. Every time you breathe in, you're breathing in air, but you're also breathing in everything else that's in the air. Smoke, pollen, dust, fungal spores, viruses, bacteria, dead bugs, who knows? Oh, come on, we've all eaten a bug or two, all right? And a lot of that stuff you just don't want down in your lungs. It's going to clog up your lungs and it's going to block gas exchange, and it can cause health problems, you know, asbestos, coal dust, cotton. The people who used to work in these big cotton gins would breathe in cotton dust all day, and they would develop horrible lung diseases. Well, our respiratory system does have a method for trying to cut down on the amount of these particulates that end up down in our lungs. And that's what's happening here. The lining of our airways is covered with cells that have cilia projecting out into the passageway. And these cells secrete mucus. They're a mucus membrane, which, as I told you, is thick and sticky, right? So you breathe in, and these particulates get stuck as they're going down. They're going to stick onto this layer of mucus. So you breathe in, and all this dust and dirt and particles and so on, hopefully, is going to end up stuck onto this mucus lining. To move it out of the respiratory system, the cilia beat, and they don't move themselves, they move the mucus. So the mucus is being slowly but steadily moved up your respiratory system until it collects at the back of your throat. And that's what you do. In fact, the urge is almost overwhelming. I put the power suggest. You clear your throat. You give a little cough. You bring that mucus all the way up into your oral cavity, into your mouth, and then, unless you're really a horrible person, you swallow it back down. You do not spit it on the sidewalk. One of these days I'm going to lose it and hit somebody for spitting on the sidewalk. You know, these big old loogies, just, bleh. Like, no, I have to walk there, all right? So you swallow it down. And now it's going to go down into your stomach, and it's going to be destroyed by the acids in your stomach.